USDA logo. United States Department of Agriculture. Meet the expert, Jonathan Frye, PhD.
Yeah, my name's Jonathan Frye. I work in the U.S. National Poultry Research Center, and my focus is on Salmonella and how it becomes antibiotic resistant.
What is the subject area of your research?
We're trying to find out, like I said, how Salmonella becomes drug resistant, antibiotic resistant, and how we might prevent that, especially on farm and in the processing environment before it goes to customers as retail meats.
Describe a brief synopsis of your research.
Well, overall, our objectives are to understand the genetics that drive this antibiotic resistance. In Salmonella, most of its drug resistance is carried on mobile genetic elements. Most of these are plasmids. They move around easily from different bacteria, and it doesn't have to be another Salmonella. It can be pretty much any bacteria. So bacteria actually exchange DNA very easily. So we can acquire these resistance genes from any other kind of bacteria they might come in contact with. So we're interested in finding reservoirs for these resistance genes out in nature or in the farm environment or a processing plant, that kind of thing. So we can see about preventing those genes from being able to transfer into our pathogens like Salmonella.
What are the main outcomes of your research?
Looking at bacteria in surface water, not just Salmonella, but also we're interested in E. coli and Enterococcus. Those could sometimes be pathogens, but they're also natural commensals. Everybody has them in their guts, and they can actually carry drug resistance and then pass that on to pathogens like Salmonella. So I had a student come to me, and she told me that she wanted to work on antibiotic resistance in surface water. I thought that was interesting. I talked with my lead scientist, Dr. Charlene Jackson, about that, and we decided that it was a good direction to go in, a little extra project, because we really weren't looking at that type of environment. So we started doing water sampling with a local group of volunteers, and we were able to take samples every quarter for several years and isolate E. coli, Enterococcus, and Salmonella from the water samples. We were surprised to find that most of the water samples were positive for Salmonella. That was a little surprising. All of them were positive for Enterococcus and E. coli. That wasn't surprising. But some of the E. coli were drug resistant, and so were most of the Enterococcus. She did a little more in-depth study on some of the E. coli, looking at the genetics of the drug resistance, and we actually found an E. coli strain called ST131, which is an epidemic strain of E. coli that's expanding throughout the world, but not really so much in the U.S., certainly not out in nature. Sometimes it's in people, usually people who've done travel internationally. So we're really surprised to find this in the surface water, so we're following up on that. Well, the important finding for me was to always look for inspiration from wherever it comes from. I was lucky to get a really bright student, Gabby Cho, to join my lab, and came up with this project idea, not one that I would have naturally come up with, but when she suggested it, everything fell into place, and it turned out to be to give us a lot of interesting information on where these resistance genes may be residing and where we can go to look for them, and maybe where we might opt to try to get rid of them.
Are there any other results of your research?
Well, we definitely want to follow up on that particular isolate. The sampling site was downstream from our local hospital. We don't know if it came from the hospital or not, or it could have come from an animal, it could have come from a human that had traveled, it could have come from a sick human. We don't know. So we're going to try to trace that back by comparing the genetic signature of this bacteria with those collected by the CDC.
What are the next steps of your research?
Just that ARS is a great place to work. It gives us the opportunity to do research that wouldn't really be possible anyplace else. I mean, I can get grants like a professor, but I also get permanent research funds, and and have an assignment to do. So I do that assignment, and I can also do little side projects like this that can turn into something pretty big later on.
USDA is an equal opportunity provider and employer. To file a complaint of discrimination, write to USDA, Assistant Secretary for Civil Rights, Office of the Assistant Secretary for Civil Rights, 1400 Independence Avenue, Southwest. Stop 9410. Washington, D.C. 20250. Or call toll-free at 866-632-9992 for English, or 800-877-8339 for TDD, or 866-377-8642 for English Federal Relay, or 800-845-6136 for Spanish Federal Relay. Music by Dexter Britton. Available under Creative Commons license from www.dexterbritton.com.